This video is going to help you to begin the process of synthesizing double-stranded RNA. And the protocol that we'll be using in this video is in vitro synthesis of double-stranded RNA for RNAi. And you'll see at the beginning there's a synthesis reaction here, and it says use PCR to generate template from PJC 53.2. Purify and elute in water. You can see the other videos for hot start PCR and DNA purification with the clean and concentrator kit for the first few parts of this protocol. Those can be used for other things too, but that's how you would start out this protocol. You would do your PCR. So going back to what we talked about when we did this PCR, you have your PJC 53.2 plasmid. We've amplified from the T7 promoter to the T7 promoter using those T7 primers to make this PCR product. And what we're going to do now is use the T7 enzyme to generate double-stranded RNA from this PCR product. So the input from the purified PCRs, so this is just PCR product from Hot Start that's been purified, that's what we're going to use as the starting material. And so this reaction is going to combine the template, which is our DNA. It needs to be between 50 and 150 nanograms per microliter, so you can check the concentration of those if you'd like. And then we're going to have high yield transcription buffer. We're going to have 25 millimolar rNTPs, so that's all of the NTPs you need to make RNA from DNA. We're going to use T7 RNA polymerase. It'll just say T7 on the lid. And this, if you've seen another video where I said people sometimes get mixed up the T7 primer and T7 polymerase, this is the enzyme, so it should be in glycerol, it should be in a smaller tube, and it should just say T7, not T7 PR or T7 primer. There's TIP, which is an inorganic phosphatase. And then there's RNasen, which is an RNase inhibitor. When you're doing RNA work, you want to be wary of RNases. So our bodies are just covered in RNases. Our bodies use them to degrade RNAs as a way of keeping away viruses or other things that could be in our environment. So if you just like put a thumbprint on something, your thumbprint would have RNases in it. That's good for our body because it's a defensive strategy, but it's bad for us as scientists because sometimes we want to make RNA and all of those RNases can be a problem. So when we're doing RNA work, we're going to use a couple of strategies to minimize the contamination of our samples by RNA. So for this protocol, it says use RNase-free filter tips. So you're going to be using all of our filter tips. All of these materials should be RNase-free, and you're going to be really careful to use RNase-free tubes. So if you've had just tubes sitting out on your bench, you don't want to use those. Grab a fresh bag. And you're going to just be careful. Like, I don't like to loom over them. I'm wearing a mask because of the COVID pandemic. But you don't want to be breathing in these samples normally because you would even have RNases in your respiratory droplets. So just kind of don't hold your breath, but don't be blowing in them either. Okay, so we're going to set up a master mix for this reaction. So this tells you all of the things that you put together. This is a 20 microliter reaction, and we're going to combine the template, the high yield transcription buffer, the rNTPs, T7 tip, and RNasen. The master mix is going to have everything but the template because you could have a lot of different templates. You might be generating double-stranded RNA from for many different genes. And so the template will be left out. We're going to make a master mix of the other reagents. And in my lab notebook page, I've already written down this reaction, and I've multiplied by a number that is the number of samples I have plus a couple, and I will set up my master mix with everything but the template, and then aliquot that and then add the template last. So I'm going to, my high yield transcription buffer and rNTPs are still thawing, so I'm going to let those thaw, and then we're going to come back and make this master mix. Okay, so I'm going to combine my master mix. I have another tube just labeled MM for master mix. And because there's no water in this reaction, I'm just going to actually combine everything in the order that it says on the protocol. There are 3 enzymes in this reaction, the T7, TIP, and RNasen, and those are going to be added last. So in any order, you could add the high yield transcription buffer and the RNTPs. So I'm going to put the high yield transcription buffer. Of course, the volumes will depend on how many reactions you have. So make sure your math is correct. So I added the high yield transcription buffer. I'm going to add the RNTPs. And remember, this is our ATP, our GTP, our CTP, and our UTP because RNA has different nucleotides than DNA. So they would be ribonucleotides instead of deoxynucleotides, and then you would have U instead of T. Okay, so I've added those 2 things to my master mix. I'm going to go to the freezer to retrieve my enzymes. So I'm to the freezer and I'm going to take out my 3 enzymes, which are, I've already put them in one blue cold block, but normally they would be back in the yellow cold block in the lab stock of reagents. So I have T7, TIP, and RNasen. I'm bringing this back to my bench, and as usual, I'm going to add my enzymes, and then I'm going to take them back while I'm setting up everything else. So I'm going to add my T7. This is our RNA polymerase, and it will be in a tube that looks like this, or something similar to that. These, remember, you're just going to take the enzyme off the block long enough to pipette it, and then you'll put it right back. I'm pipetting into the liquid, and it's a little bit viscous. It's in glycerol, so I will mix them well in a moment. I'm going to add TIP, which is an inorganic phosphatase. This is not necessary for the reaction, but it facilitates and improves the efficiency of the reaction. I'm trying to make sure I didn't have a bubble in there. Also, it'll be viscous since it's an enzyme. And then last, I'm going to add the RNasen. This is RNasen plus. Sometimes we'll have regular RNasen. And that will be added last. So I'm going to take these 3 enzymes back to the freezer, and then I'll be right back. Okay, so I've added everything for my master mix. I'm going to just flick it a little to have it mix up. And then I'm going to add my master mix to each tube. So if you look at the reaction volume, the total volume is 20 microliters and the template is 10.5. So you'll add 9.5 microliters of your master mix to each tube, and then come back and add the 10.5 microliters of template DNA to each tube. So first I'm going to add my master mix to each tube. I've labeled all of these tubes DS for double-stranded RNA, and then something about the gene name. You can number them, you should have the date, and so on. So now I'm going to pipette the master mix into each tube. I'm just doing this on a rack on my bench. You can also, occasionally if you need a whole lot of double-stranded RNA, you can double these reactions and then double everything that follows in this protocol. So say you needed, you found a really interesting gene and you need to knock it down for multiple different follow-up experiments. You can set this up in multiple tubes, but also you can double the reaction volume. Okay, so I've added my master mix to each tube, and now I'm going to add my template DNA to each tube. And you're going to need to use a different tip for each one because your templates are often going to be different. So I'm adding the template DNA to each tube. I'm going to give them a little flick to mix, and then I'll put them in the incubator. So once you're finished with your reactions, you're going to go to one of these little machines that are around the lab. You may need to turn it on. You're going to set the temperature to 37 degrees once it's warmed up, and you're also going to put your samples into the machine. So here I'm putting the samples in. And then I'm going to check to make sure this is, I'm going to do my reactions at 37 degrees overnight. So I'm going to make sure this is set at 37. Your high yield transcription buffer and RNTPs can go back into a freezer box. Any PCRs that you have left over can go into a freezer box, and any tubes that you may have that are empty, if you've used up your master mix or if you've used up the DNA from your PCR, those can just go in the trash. So we'll let this go overnight, and then we will resume the protocol with DNase treatment and cleanup. So I'll see you then. All right, so we're back with the second portion of the in vitro synthesis of double-stranded RNA protocol. I have allowed my transcription reactions for the double-stranded RNA to go overnight at 37 degrees. In the protocol, it says you can leave this for 5 hours, and that's fine. So there may be sometimes when you're in a hurry that you want to do all of this in one day, but often I will do the next couple of steps the day after I've set up the synthesis reaction. And so if you imagine what's in this tube, we had our PCR product, and then we added the reagents to make this double-stranded RNA, so synthesizing the RNA from the DNA. In our tubes then we have the template PCR product, we have our double-stranded RNA, and then we have all of these enzymes and buffer reagents that we used to make the double-stranded RNA. So what we're going to do today is to first DNase treat the samples, and this will get rid of the DNA template. And the second thing we're going to do is clean up. So we're going to precipitate the nucleic acid out of this to separate out those enzymes and other molecules that are in our tubes. So the first thing we're going to do is our DNase treatment. The protocol says that you can just add 1 microliter of DNase to each tube, and that is one way to do it. I actually sometimes notice that there is evaporation, and so what I like to do is actually add a combination of water, high yield transcription buffer, and DNase, and make a master mix of those 3 things, and then add 10 microliters of that mix to each one. So if you choose to do that, you're going to, for each sample, combine 8 microliters of DEPC or RNase-free water, 1 microliter of your high yield transcription buffer, and 1 microliter of DNase. You can multiply that by however many samples you have, add a couple for overage, and then make a master mix, and from that master mix you'll add 10 microliters to each tube. So I'm going to make my master mix. I have 12 samples here, and remember, for this protocol we're going to be using these filter tips, which are RNase free. So I have in front of me a tube for my master mix, my high yield transcription buffer, and then my double-stranded RNA synthesis reactions that I've taken out of the 37-degree incubator. So I'm going to combine the reagents for my DNase treatment. First, I'm going to add water. I think, as I mentioned before in one of these videos, I usually add water first. If it's one of the ingredients, it usually is the primary ingredient with the highest volume, so I like to get that in there first. Next, I'm going to add the high yield transcription buffer, which was the buffer that was used for the transcription reaction. When you're, I should mention, when you're thawing these buffers, make sure that they're completely thawed, there's no ice left. And if you see any little precipitate in the bottom, make sure that's totally resuspended before you proceed. And then I'm going to go get the DNase from the freezer. I will take you with me. So my bay is closer to the door, and the common reagent freezer is in the other side of the lab, right here. And again, this is one of the enzymes we use, so we want to keep it on a cold block and always get it out right before we need it, and then put it back right after we use it. The DNase that we're currently using has an orange lid, and we'll say RQ1 DNase on the top. So I'm also going to add my DNase to the master mix that has water and high yield transcription buffer in it. It's a little viscous. It's in glycerol like all the other enzymes are, so just make sure you pipette accurately. So I'm going to just flick this a few times to mix it, to make sure all that glycerol is resuspended. And then I'm going to take the DNase back, and we'll resume in just a moment. So after I've combined all the reagents of the master mix, I'm going to pipette 10 microliters of that into each tube. And I'm just pipetting into the liquid and stirring a little bit with my tip as I pipette into the tubes. Generally, you don't want to pipette nucleic acids up and down. The shear forces that can happen when you do that, either for RNA or DNA, can rip apart the molecules. So in general, you don't want to sit there and pipette up and down if there's a nucleic acid like DNA or RNA present. There are other molecules or buffers for which that's not a big deal, but for nucleic acids, I would avoid it. So after this step, you'll notice that some of your tubes seem to have a little bit of liquid on the sides. Remember that this step is meant to remove all of the DNase from the solution. So if you had, like for example, this tube has a little bit of a droplet on the side. If you flick the tubes, you can often get most of the liquid consolidated in the bottom. And then what you may choose to do is to spin down these tubes just a pulse in the centrifuge to make sure that if there are any droplets up on the side of the tube, that they are pulled down to the bottom for the incubation. So I'm going to pulse centrifuge these, and then I'm going to put them back in the warm block for 15 minutes. And after that 15-minute period, we're going to precipitate the double-stranded RNA that remains in the tubes and proceed with the protocol. Okay, so it's been 15 minutes. I'm going to get my samples out of the 37-degree block. You can turn the block off after you're finished, and you can put all of your samples on a tube rack to bring back to your bench. Next, we're going to add the materials to precipitate the remaining RNA. Remember, the DNA is now gone, so you have all of the reaction reagents and enzymes, and then the only nucleic acid left should be the double-stranded RNA. So we are going to add 3 things to these tubes to precipitate the DNA. All of the proteins, like the enzymes, will stay in solution, and this will allow us to purify the RNA from everything else in the tube. So the first thing we're going to do, as this protocol says, is add 80 microliters of RNase-free water. You may have depth seawater. I'm going to add 100 microliters of 5 molar ammonium acetate. And if you look at the samples when you add that, it might become opaque. And then you're going to add 400 microliters of 100% ethanol. And we have some 200 proof ethanol that we use for molecular biology experiments. So I'm going to add these things to each tube. I'm going to use the RNase-free filter tips, and I'm going to just do maybe 1 or 2 of them, and then you can see kind of what things look like. So the first thing I'm going to add is the Depsy water. This won't do anything. The water itself won't do anything. I'm just going to do two of my tubes and then pause for a minute to do the rest. I'm then going to add the 5 molar ammonium acetate. If you notice that my volumes are a little bit different from yours, I'm actually doing double reactions, so just in case you're wondering why I have a pipette that's not what you expect. You should be adding 100 microliters of the 5 molar ammonium acetate. And then you'll be adding 400 microliters of the ethanol. And I don't really like to pipette in and out of that big bottle since I use it for quite a long time. So I just have a second bottle that I keep the same ethanol in for this purpose. So sometimes you'll see a little bit of like swirliness after you add the ammonium acetate. It's kind of a little bit swirly. And you may notice that as well when you add the ethanol. And that's the nucleic acid precipitating. As with a lot of things that we do, I might use 1 tip if I'm able to pipette from the ethanol into all of these things without touching anything or splashing anything. If you ever feel that you have touched something or may have contaminated it, your pipette tip, you don't want to go back into your ethanol or your ammonium acetate or things like that with double-stranded RNA. So maybe when you're starting out, you might use separate tips for everything, and then as you become more experienced, you can use 1 tip for a whole set. Just be very careful and be very aware of where your tips are. If you invert your tubes, you can kind of see, I noticed it in this one, some like kind of chunky, clear material moving around. I can notice it again in that one. And that's the precipitated RNA. So what we're going to do is I'm going to pause the video so that I can add these reagents to the other tubes, and then we'll put these tubes in the freezer together. Okay, so when you're finished adding the precipitation reagents for each one of your tubes, you can invert them a few times as I showed you, and you may or may not see a little bit of material. You can kind of see it in these tubes moving around. I think it looks sort of clear and gel-like, but sometimes it will look a little bit white or cloudy. So this, as you can see, will already start precipitating the material, but you can also facilitate this by putting your reactions in the freezer. So we have several -20 freezers around in the lab. I have one right here that's by my bench, so I'm going to use it. And you want to I just stick the whole rack in there. And then set a timer for an hour at least. So since I'm putting it in a rack, and that rack is at room temperature, I'm going to start a timer and I'm going to wait for about 1 hour and 15 minutes. You can also have this precipitation incubation go overnight. So if, for example, you set up your synthesis reactions and then you wanted to come in and just do the DNase and precipitation steps, you could do that. And then you could resume the cleanup, which is the centrifugation, and the next few steps the next day, or even, you could wait until after a weekend. You can wait a few days. During this incubation, you're free to do other things. You might catch up your lab notebook, but there are a couple of things that I would recommend doing. if you are going to proceed with the cleanup at the end of the incubation. The first is that I would recommend chilling the centrifuge. So we're going to use this centrifuge that's by my bench right here. And we want it to be at 4 degrees. And it isn't typically at 4 degrees. So if you want to set it that way, you can chill it in advance. It takes a little bit of time. So I'm going to, sorry, I'm like trying to close it and look at this. Okay, so I've closed the centrifuge. I'm going to reset the temperature to 4 degrees. This is just to keep those samples cold. And then there's a button that says fast temp. And if you hit fast temp, you need to make sure that lid is on the rotor. But this centrifuge will go through a spin to cool down to 4 degrees. That takes about 13 minutes. So it's a great idea to set up that cold centrifuge before you're ready for it. If you waited until the end of the incubation to cool the centrifuge, you'd have to wait even longer. The other thing you can go ahead and do is, if you plan to pour and run a gel after your purification, you can pour that gel during the 1 hour incubation as well. So I'm going to go do that. The other things you can do are just make sure that you've put everything back. You should have put away the high yield transcription buffer right after you'd used it, but just in case you didn't, put that back in your freezer box. And then the next steps will be to continue this protocol with the centrifugation steps and the annealing. And I will be back in a moment. Okay, so at this point in our protocol, my double-stranded RNA has been incubating for over an hour. And the next few steps will be to centrifuge that precipitated double-stranded RNA as per the protocol, to pull that material down to the bottom of the tube and wash it off and then resuspend it in water. So the first thing we're going to do is to spin our samples. So we'll come and get them out of the freezer. You may be able to see a precipitate at the bottom. So like a little whitish clear pellet. I'm going to put these in the centrifuge. So I'm going to open it up. Take off the lid. It's a little hard to do one-handed. And then I'm going to put in my samples. Again, every time you use the centrifuge, you want to make sure that the samples are balanced, meaning that you have tubes on each side, and you can use just a tube that has water in it if you have an odd number of tubes if you need to. So I've put my samples just across from each other in the centrifuge. I'm going to now close the lid, make sure that it clicks. The first spin is for 15 minutes, so I'm going to increase this to 15 minutes. And this is still at 4 degrees, and I'm going to set this at maximum speed. If you have forgotten to cool down the centrifuge, you can cool it down and leave your samples in the freezer until it's cooled down. And then you're going to hit start. And then this spin will last for 15 minutes. At the end of that spin, I'll be back and we'll talk about the second spin. Okay, so I've removed my samples from the centrifuge after the first spin. And I'm not sure if you can see, there's kind of a whitish clear pellet on each tube. You can see it there. I always put the hinge on the tube outward so that I know that the pellet will be on that side of the tube. Just another little tip. So what we're going to do next is remove the ethanol and ammonium acetate mixture. You can have a waste container, like either just an empty tube or a one of those tri-corner beakers for your liquid waste. So what you're going to do is remove the liquid without disturbing the pellet. So you don't want to suck up the pellet. You want to make sure you can see it. and we're just going to pipette away from it. Remember, I'm doing a double reaction, so I may have a little bit more liquid in here than you have. What should be left is just that little pellet on the bottom. You may see some of the pellet up the side too. You want to remove as much of the liquid as you can, although that's more important after the next step. And I will say you want to make sure that the centrifuge is closed while you're doing this so that it stays cool. So just make sure that you've done that. And I'm just being very careful not to disturb the pellet. If you can't see your pellet, you want to be a little bit more careful, and you might leave a little bit more of the liquid at the bottom in this first step. If you don't see a pellet at all in either step, that might be a sign that the double-stranded RNA synthesis didn't work. But you can keep moving forward, and sometimes there's a little bit of a pellet that you just can't see. I think that often these pellets will appear to be kind of clear or glassy. They're kind of gooey, sort of like a gel-like consistency if you accidentally try to pipette one up. Occasionally they will instead be kind of white and chalky, and we found that for the most part there's no difference in the quality of that double-stranded RNA or the ability to induce knockdown of genes. So this may have something to do with the buffer conditions just varying, or other reaction conditions slightly varying, and you get a different pellet. Anecdotally, I always am happier when I get a glassy pellet, but I don't think it really matters too much. We've had success with both types. It's reassuring when you get these pellets, I would say. For those of you who have done riboprobe synthesis before double-stranded RNA synthesis, riboprobe synthesis causes a very small white pellet, like almost invisible. So if you've gotten used to that first, this double-stranded RNA pellet will be really big and impressive by comparison. In contrast, if you're doing this first and then you're going to do riboprobe synthesis later, the pellet will probably be much smaller for that. Okay, so now I have all of my samples, I have the pellets at the bottom, and there might be a little bit left of our solution, but not a lot. I've gotten rid of as much of it as I could. And I'm going to then, and I was using a separate tip for each tube, you may have noticed. Now I'm going to use 70% ethanol, so this is made from the ethanol that we used before, and depth sea water or RNase-free water. I'm going to add 500 microliters of 70% ethanol to each tube. And the purpose of this step is to wash the pellets to make sure there are none of those salts that were from the buffers, to rinse off all of the enzymes and things like this. So this sort of washes the pellet. So the only thing that should be in the pellet is the nucleic acid and not those other components. Again, I'm using one tip because I'm being careful not to touch anything. If you touch anything, switch your tip. Okay, I'm going to invert my tubes just twice or 3 times. You'll see that the pellets will become sort of dislodged. They might also change color a little bit and become a little bit more white and opaque. You can see that one there. And then I'm going to spin for 5 more minutes at maximum speed at 4 degrees Celsius, and then I'll get them out for the last step. So after the second spin, you should again see your pellet on the side of the tube. And this time we really want to remove all of the supernatant so that we can allow these pellets to actually dry. If you left ethanol in these pellets, they wouldn't dry, and that would interfere with your downstream applications. So first I'm using my P1000 tip to remove as much of the liquid as I can. Then I'm going to spin just a pulse spin in the centrifuge. And then use a smaller pipette tip to try to get all of the liquid out. So I've done 2 of these. You can see I've gotten almost all the liquid out. I'm going to do that for the rest of these tubes. I'm going to take my tubes to the centrifuge, just spin down just for 5 or 10 seconds is fine, and then I'll bring them back for the final part of the spinning process. So in the centrifuge I was using, you can use the button that says short if you wanted to just do a pulse spin. Now what I'm going to do is remove the rest of the liquid, which is now at the bottom with the pellet. Depending on how good of a job you did with removing the supernatant, you can often set a P10, a P20 that has a nice sharp tip to remove this supernatant. And it might take a couple of pipetting actions. So at the end you'll just have the glassy pellet at the bottom. So if you did a little bit less well and you have a little bit more volume, you might, as I mentioned, use your P20. Again, you don't want to disturb your pellet or suck up your pellet, but you want to get all of this liquid out. So you can see that here. You are going to leave these tubes open once you've removed the supernatant. And the reason you're going to do that is we're going to just leave them on our bench top for 5 minutes and allow these pellets to dry. This will allow any excess ethanol to evaporate. And ethanol can interfere with downstream applications. It can cause problems if you try to feed double-stranded RNA that had too much ethanol in it, and so on. So we always want to just make sure these pellets are completely dry. So I'm going to finish removing the liquid, and I'm using kind of just eyeballing whether I need to use the P20 or the P10. You should see the liquid kind of go up into the pipette, and then, you know, there's a bubble because there's no more liquid for the pipette to remove. And I'm putting these tubes down to the bottom of the down to the bottom of the tube, but beside the pellet. So the pellet is kind of angled, and I'm putting this on the bottom of the tube, but on the side away from the pellet. And with these tiny tips, and if the pellets are this big, you probably won't be able to suck them up anyway. Okay. So I have all of my tubes, I have the lids up. As we mentioned, you don't want RNases in your RNA. So what I do is I just take one of these chem wipes and drape it over the samples. This is just to keep dust from falling into the samples while they're drying. And I'm going to set a timer for 5 minutes. This will allow the double-stranded RNA, any remaining ethanol, to evaporate. What's left will be our purified RNA. At the end of that 5 minutes, you should be able to see that your pellets are still there, but there's really no liquid left. You've removed all of it and kind of given it some bonus time. You might see a little bit of kind of clear or glassy residue up the tube. That can be part of the pellet too. So the next thing we need to do is resuspend your double-stranded RNA in RNase-free water. So I'm using depth sea water, but we have other RNase-free water too. That would be fine. And then I'm going to add 30 microliters of RNase-free water to each tube. Again, I'm using just one tip, but I'm not touching anything. And you can decide if you want to switch tips or not. Okay, so I have my tubes in the rack. I've added the water. I'm going to cap the tubes. And at this point, the pellet should be a little bit whitish or opaque. And you want to flick the tubes until the pellets become dislodged and sort of up in the liquid. And it will take a moment or two for the double-stranded RNA to resuspend. So what you can do is just set a timer for 5 minutes and then come back. If you still see one of those pellets that hasn't fully dissolved, you might give it a few more flicks. That's what I'm doing to just dislodge the pellets from the bottom. Start my timer. Give them 5 minutes or so, and then we'll be ready for annealing. The next step of this protocol, so at this point you've purified your double-stranded RNA, and you can change the volume of water you add. So if you don't think you have very much, you could add less. If you have done double reactions or you have really big pellets, you might need to add more. The next step, number 5, is annealing. And although you've made double-stranded RNA, i.e. RNA from both strands of the PCR product like this, that doesn't mean that all of that double-stranded RNA is perfectly paired with its antisense strand. You know, you could have bits of RNA that are paired with more than one strand of RNA and things like that. And so what this annealing process will do is you will heat your samples up to 95, which will melt all of those complexes apart. And then by slowly lowering the temperature, you will encourage pairs of RNA to bind together specifically, so you'll truly have double-stranded RNA. This can be done more than one way. So some people like to resuspend their pellets, spin them down, and move them into PCR tubes. Let's look like this. You need to label them really well if you're going to do this. You could then take your reactions, cap them, and put them in the PCR machine, where there's a program that will take your samples through these different temperatures for the indicated time. I prefer to just leave my samples in these 1.5 ml tubes. After this is, after my pellets are resuspended, I will spin them down, just a pulse spin in the centrifuge, and then I will put them in the hot block that I used for my 37-degree incubation, which I have set at 95 degrees. And so this protocol says 95 degrees for 3 minutes, 75 degrees for 3 minutes, and 50 degrees for 3 minutes, and then allowing to cool at room temperature. Because the heat block takes a few minutes to get from one temperature to the other, I actually do more like a 5-minute incubation, or I wait till the temperature is at the necessary temperature and then start a 3-minute timer. So it probably takes a little bit longer to do the annealing in these tubes, but you save yourself that step of having to transfer them to PCR tubes. So you can decide which of those you prefer. You can try them head to head and see what you think works best. So as soon as I have cleared samples with no pellets, I'm going to put them in the incubator. I'm going to do my annealing steps. Actually, I'm going to pulse them down in the centrifuge and then put them in the incubator for the annealing steps. And then I will take them out and talk you through how you can run a gel to quantify the double-stranded RNA that you've created. Okay, so I finished with the annealing process. I've taken my tubes out and spun them again in the centrifuge to make sure all the liquid and condensation, which can happen during the heating process, gets back down to the bottom. So I have my purified double-stranded RNA here. I've also gone ahead, the next step is to run this sample out on a gel. And I've made another video of pouring and running agarose gel, so I'm just going to tell you what I do differently for double-stranded RNA than what you saw in that video. One thing is, instead of using orange G loading dye, I'm going to use a formaldehyde load dye. This is in the common reagents section in the freezer in the cold block. This doesn't have to be kept on a cold block, but you want to just get it out when you need it and then put it back. I also have made myself a little parafilm setup so that I have one of these little dimples for each sample, and I'm going to use that first set of dimples to make a 1:10 dilution, and then I'm going to use the second set of dimples to actually make the sample that I'm going to load. So what I'm going to do first is to pipette 9 microliters of RNase-free water into that first set of dimples. So I'm going to do that quickly. And the reason that we have to dilute this double-stranded RNA before we run it on a gel is that this is a very concentrated sample. If we were to just run out 1 microliter of the double-stranded RNA, and you're welcome to try that, it will be really smeary and super bright, which tells us that we have a double-stranded RNA and a lot of it, but it doesn't tell us exactly how much. So what we're going to do is make this 1 to 10 dilution so that we have a fainter band that we can actually compare to the latter to estimate the concentration of the double-stranded RNA. I just noticed my chair squeak, so sorry about that. So I've put 9 microliters of water in the first set of little wells that I made. And then I'm going to pipette using an RNase-free tip, 1 microliter of each sample into the corresponding droplet. Make sure that if you have multiple samples, you've gone ahead and put them in the right order. You don't want to have sample 1, 7, 3, whatever. You want to have 1, 2, 3, 4, 5, 6, 7, 8, 9, and so on. And make sure that you're paying a lot of attention. You don't want to pipette 2 of these microliters into the same spot or anything like that. So I'm using a different tip to pipette 1 microliter of each of my 12 samples into the corresponding droplet of water. Okay, so now I have my 12 droplets in my top row, and these have a 10 microliter sample in each, and that's a 1 to 10 dilution of my double-stranded RNA in RNase-free water. In the second set of dimples, I'm then going to add 8 microliters of, or 7.5 is fine, of RNase-free water. So I'll do that really quickly. And the second row is the set of samples you are going to load. You're going to actually get rid of the top samples. Those are just a spot for you to make the dilution, and then you're not going to run them on a gel. Okay. After you've added your nuclease-free water to your second row, you're going to add 1 microliter of the formaldehyde loading dye into each little droplet. And I sort of estimate this. I load up a tip with 10 microliters, and then I add this to each. It's actually probably more like a microliter and a half. Okay. So you just want them to be blue. They're going to look a little green on here because I have a yellow rack. And then the last step is to take 1 microliter from your dilution and put it in the loading dye. So let's see if I can angle this so that you can see it. So I'm going to take my first microliter from my top left dimple, and I'm going to move it into the blue dot below it. And then I'm going to do the same thing for each of my other samples. I'm going to use a fresh tip with each of these because I don't want to contaminate one of the samples with the other double-stranded RNA. Be careful when you're pipetting 1 microliter, it's not very much, so just make sure you really are paying attention to how much volume is coming up into your tip. Let's see if you can see it there. Okay, so now I have in the blue droplets the water, the formaldehyde loading dye, and my 1 microliter of my 1 to 10 dilution. Before I set up my gel, I'm going to take this formaldehyde load dye back to the freezer. I'm also going to put my double-stranded RNA samples into my personal freezer box, just because I don't want to forget about those and have them degrade while I'm working on my gel. And I'm going to load a gel with one lane of the ladder. I'm going to add 2.5 microliters. You can add 3 microliters if you want, but just be really careful to note how much you add. That's the hyperladder 1 kb. I'm going to skip a lane, and then I'm going to load all of my RNA samples. I'll show you what that gel looks like after I've imaged it, and I'll talk you through how to quantify how much double-stranded RNA you have. I'll mention that the formaldehyde load dye is a little bit better for running out RNA, because it helps to undo some of the secondary structure that can form with RNAs, either single-stranded or double-stranded. But that's why we use this instead of the orange G for this and for riboflavins. All right, so I will show you my gel once we're finished and talk you through quantifying how much double-stranded RNA you've created. And then my samples, each one of these is 1 microliter of 1 to 10 dilution. And you can look up one of these ladder information cards online. This is just one that came with one of these tubes a long time ago, but you can find this online, and you can see that this bright band at 1 kb, there's 100 nanograms of DNA in this band if you load 5 microliters. I loaded half that, so this bright band right here is 50 nanograms of nucleic acids. So if I decide that my band here is about the same as that, then that means that this band is about 50 nanograms as well, and that since this is a 1 to 10 dilution, this is really 500 nanograms per microliter. You can see this might be less, but I would say maybe these two are about similar. So this is really a way of estimating the concentration that you have. And you'll see that there's some smearing. This is where just not all of that RNA anneals properly. And you can re-anneal a little bit. Sometimes you'll have a stronger single band than this, but a little bit of that smeariness is normal for double-stranded RNA.